Well, maybe we'll get started. Welcome, everybody. This is, I'm David Brainerd. I'm the director of the Penn's Vision Research Center, and we've done this periodically in the past where we take one of the times scheduled for the Vision Science Seminar just to give an overview of the Vision Research Center and our cores and make sure everybody knows what we have available. This seemed like a timely time to do this for the reason that the Vision Research Center is primarily supported by a P30 core grant from the National Eye Institute of the National Institutes of Health. Probably most of you know that. And those are funded on and awarded on 5-year cycles. So, and we have one of the longer standing ones. I believe we're in year 46 of this grant, with the first PI being Paul Liebman, followed by Peter Sterling, and then me. And we're now in year 1 of the 5-year grant cycle. So we have funding for the core grant through 2025. And we have some updates to our modules, which is always good to tell people about. I'll also point out, although that we have at Penn 2 National Eye Institute training grants, a T32, which Diego Contreras is the PI of, and a K12, which Josh Nanev is the PI of. And those are, we loosely try to stay in touch between what's happening with the core grant and the training grants. In fact, the Vision Science Seminar is funded in part by funds from the T32 and in part by funds through the Vision Research Center. The main thing that the Vision Research Center does, at least through the P30, is provide support for vision research on campus. And our goal really is to support individual labs and things they do by having sensibly designed core modules, which we think about every 5 years as we do the renewal. We also sort of aim more generally to foster collaboration on campus in vision research. We have a fantastic and very broad vision research group here at Penn, really covering the full scope of vision science, both basic research and translational. And so it's sort of one of our missions is to keep track of who's doing what and to think about how to foster communication and collaboration across the scope of our field. The Vision Science Seminar is one way we do that. And we've, at least when we get back through the pandemic, we will have, again, start up the Vision Dinners, which are co-sponsored with ophthalmology. Many of you may remember these. We typically have some talks and then a nice dinner. I do that once a semester where we catch up on what some of our community is doing and then have a chance to say hello in person. And all I can say is I'm really looking forward to the time when that's possible again. Because we have the grant renewed, some funds I was kind of bankrolling that are unrestricted funds and keeping in reserve in case it took a second try to get the core grant funded. We're going to use to have some seed grants for new collaborative research, and we will send out on our mailing list to the PIs information about that once we've finalized the scope of the program. And we're also thinking about ways we might structure some workshops or retreat or something, possibly virtual, that would be focused on specific topics where we think there might be opportunities for collaboration. Just in terms of what we're going to do today, the module directors are going to give an introduction to our core modules and what they do. I'll say that the National Eye Institute does require us to give priority for module support to faculty funded by individual R01 grants from the National Eye Institute. That's a requirement of the P30 program. But then we also have tried and been largely successful at extending that support, particularly to new investigators, pilot projects, and then as long as we have capacity to vision-related research generally on campus. The main thing that we do with our budget actually is to support staff, and you'll hear a bit more about that. And the final thing I'll mention is that there's an executive committee, the names of which are here, and there's a handout in the chat, which you can download for reference, which has our names and contact information. So if you ever have any questions, ideas, or concerns, you can let me and the executive committee know. If I can, oh, there we go. So just a quick overview. We have in this cycle 4 core modules: instrumentation, a new module called scientific transparency, which incorporates the previous module, the image analysis module. We have a shared imaging electrophysiology module and biostatistics. Each of these modules has one or more primary contact persons listed in that sheet in the chat and also on our website, which is here. And you can contact the primary contact person if you have a project or a potential project that might benefit from the module. We also support the use of 2 real-time PCR machines, and there's contact information about how to find out and get access to those on a sort of self-serve basis. And with that, I'm going to stop my share and turn it over first to Jim G, who will talk a bit about image analysis, and then we'll go through the other modules. And with luck, we'll have time at the end for any questions or discussion.
Can you see my screen? Great. Thanks, happy to kick things off. The image analysis submodule of the VRC is focused on supporting Penn Vision investigators interested in pursuing quantitative analysis of images acquired for their research. The module is ably led by Min Chen and leverages the image processing and analysis expertise and resources available through the Pixel Lab that I direct to offer customized solutions for automated annotation of imaging features or structure, characterization of imaging biomarkers, spatial and intensity normalization of population data for group level analysis, and pipeline and processing frameworks for data-intensive imaging studies. Our efforts have covered a wide range of studies involving, it looks like my slides are stuck here. Is that the case?
Yeah, you're still on the first slide. Maybe try hitting escape and sharing them again.
Oops.
I'm not sure.
It looks like my screen, is it moving now?
I don't think so.
Brilliant.
Not yet.
Do you see a couple of examples on the screen?
Nope.
Maybe try stop sharing and restart, or perhaps just show your slides in the full screen mode.
Yeah, why don't I do that? Let me try this again. I'm just going to share my desktop. Can you see the slide now?
It seems to be thinking. It says you've started screen sharing, but it hasn't popped up.
Still not working? We could do this. I could jump in and do my little bit, and you could troubleshoot there for a minute.
Okay.
That sounds good. So Jeff, yeah, go ahead and do that.
Yeah. Hey everybody. So Jim and I sort of are co-components of this module on scientific transparency and image analysis. So when Jim gets control of his screen and starts showing you some stuff, he's going to demonstrate some of the amazing analyses that he and his group, and Min Chen in particular, have implemented. And what I wanted to talk a little bit about is tools that we are developing here at Penn for creating a platform to store and organize your data, and then to be able to reproducibly apply those analyses to your data so that your knowledge of how to do this stuff is very well documented and persists long after, for example, Min is hired off to Google and goes and works someplace else. So, you know, all of us collect data a little bit like this: MRI data, OCT images of the retina, you know, in Jessica Morgan's lab, AO imaging of the cone mosaic. And we have analyses that turn it into some kind of results. And those analyses have all sorts of pipelines. So there might be labeling of which subject is which, and you'll have a spreadsheet that says who they are and what their refractive error is and something like that. And then you'll have some software tools that get you from the raw data to the results. And the thing that all of us might be able to do a little bit better on is having a systematic way of capturing and preserving that data so that it doesn't decay. I don't know about you, but for some analyses I used to do, a year would pass and I'd go back and try and do it. And I'd say, oh gosh, what exactly were the inputs to this? And where did I put the Excel spreadsheet that lists exactly which subject was which? And it's tough to reconstruct. And so could we do a little bit better to build a platform to kind of allow this to be more reproducible? That's what this module is about. I'm going to focus just on the MRI data to start, but as I'll mention at the end, we're trying to extend this to other forms of retinal imaging. So these are tools to support data storage, analysis reproducibility, and collaboration and distribution. So focusing on MRI things to start and providing support for those VRC investigators who are making use of MRI imaging data to study the visual system, we've been using something called the Flywheel platform. This is a cloud-based commercial platform for storing and analyzing data. It is secure, it is covered by, it's IRB compatible in ways that I'd be happy to describe, but let me just mention a couple of things that it does. It allows for in an automated manner, all data that are collected on research MRI scanners at Penn get slurped up into the Flywheel system and automatically organized by lab and by project within the lab. And that data is now stored in the cloud and is secure and is backed up redundantly. There's an entire system that's built within this Flywheel platform to now curate your data. So to assign subject IDs and some demographic information, to attach different notes and information and results, and then to analyze the data and then to access that data either through like a very friendly web-based GUI, so a click and point, or through software tools that can access the database through an SDK. So you can write code that can access it, or you can go click and point through a web browser. And then within this platform, once your data is up there, you can process it using so-called gears. These are just encapsulated analysis tools. And there are analysis tools like this for many of the current open source popular ways of analyzing fMRI data. And you can make your own tools. And if anything is analyzed within this platform, it's provenance tracked. You know how the data got in there, how it was analyzed, and you can always reproduce how it got there. And then finally, it's really easy to collaborate. You can create accounts for people at other institutions. They can log in and access this data too. You can share these tools and pass them between different labs and between different institutions, so that if you develop something new, you can disseminate it to others. This is just a screen capture of what the web-based interface looks like for looking at some MR data, where you've got like a point and click interface with lists of subjects and different kinds of measurements that were made on them. And then there's web-based browsing tools that you can inspect the data while you're up in there, and then ways to analyze the data and do quality checks and sort of display the results of that, all in a very graphically friendly way. So we have a number of vision-related tools that have already been developed on the platform, which support things like retinotopic mapping and segmentation out of the visual cortex and the lateral geniculate nucleus, and doing DTI tracing between these structures. These are all tools that are available on the platform and that you could apply to data that you collect. And we are starting to bring retinal imaging data on the platform as well. And let me say that the VRC supports the cost of storing retinal imaging data on the platform, and can also subsidize the cost of taking your own analysis tools and then packaging them up and making them available on here. So I'll stop there and hand it back over to Jim.
Jim, you want to try again? Without something, I'll just say that, you know, if you have questions, we'll hope we have time at the end for a little discussion. And if not, each of the module directors just be happy to chat with you in more detail about what their module does. Great, we're seeing it, Jim.
And I think you're still muted, Jim.
Oops. Can you hear me now?
Yes.
Great. So apologize for the technical difficulties. Hopefully just showing this will work. So our efforts have covered a wide range of studies involving numerous different ophthalmic imaging modalities, including fundus color photography, optical coherence tomography, adaptive optics scanning laser ophthalmoscopy, conjunctival photography, and cellular microscopy. And just to give you a few examples of recent collaborations, the bottom figure is from a project led by Dr. Vatini Banya, where we developed an automated machine learning-based approach to detect the lissamine green staining in the conjunctiva. Our results showed accurate detection of the stain that correlated well with manual ordinal rankings suggesting its viability as an automated tool for the study of dry eye disease. On the top right is a figure from an ongoing project with the POAG study under Dr. Joan O'Brien's leadership, where we helped develop a new approach for studying the cup to disc relationship in color fundus photography from patients with glaucoma. A method explores the radial measurements of the cup and disc and characterizes this radial pattern to help describe properties of the optic nerve head, including eccentric and symmetric cupping, as shown here, as well as notching. Here are a couple of figures from work with Dr. Jeff Aguirre, where we studied OCT images from a normative population as part of the Human Connectome for Low Vision and Blindness
Again,
project.
Jim, it doesn't seem to have advanced.
All right, is it frozen again?
That's how it is on my view. I'm still seeing the first slide of images with the
thumbnails on the left are very impressive, though.
Yes.
Well, maybe you could just say a few words about it, and
Can you see us now?
No, it's frozen.
no.
It's still frozen?
Yeah.
Yeah.
Why don't you just say a few words about each of those projects, and then you can maybe offline make a PDF of the image data and pop it into the chat or something. People can look at it later.
Yeah, I apologize about this. It's too bad. Anyway, for Jeff's study, we developed a method for combining multiple OCT acquisitions to generate an ultra wide field representation of each subject's retina, and this allowed us to relate up to 50 degrees of eccentricity of the ganglion cell layer, together with measurements of myopia and magnetic resonance imaging-based volume measurements of the optic chiasm from the same subjects. I would now be moving on to a very beautiful slide of works that we've done with Dr. Jessica Morgan to help automate the analysis pipeline of high-resolution adaptive optics SLO images of the cone mosaic. Due to the very high resolution of this modality, each individual image only covers a small field of view, and so adjacent images have to be montaged into a single collection before we can accurately quantify the cones. And to help with this process, we developed an approach using feature matching to automatically detect and align adjacent overlapping images in both cross-sectional as well as longitudinal data. Now, prior to this work, these montages were performed manually, and we estimate that the software saves about 8 to 10 hours of manual labor per data set collected. And just as a final note, and this piggybacks on what Jeff had just mentioned, the analysis methods developed through our module are all open sourced and designed to be shared and used by the broader vision community. So for example, I have links to code repositories that we'll make available after the presentation. And as well as you've seen, much of our work has not only advanced the vision research studies they support, but have resulted in additional publications of the developed novel methodologies themselves. And these, in the best case scenario, have helped win new grant funding based on both the preliminary data and new methods generated by the module. Thanks, and I apologize for the technical difficulties.
Thanks, Jim. I think even with the, as Jeff said, even with the thumbnails, we get a sense of those images and the kinds of things that the image analysis module can do. And again, as I said before, just get in touch with Jim or Min if you have a custom image analysis problem you would like them to consult on. So Claire, are you ready to go on the shared imaging and electrophysiology module? Jim, you'll have to unshare. Great.
Okay. Can you see my screen?
Yes.
Okay. I just want to introduce to everybody some of the many techniques and tools that are available through the imaging and electrophysiology module. In a sense, we are a large module, and we support large imaging and electrophysiology apparatus, but importantly, with trained people to help you. these approaches are complicated, and so the expertise that comes along with the pieces of equipment is much more important than just having a fancy piece of equipment. What we really want to be able to do for our VRC members is to act as a collaborative work hub, and to help you understand different ways in which you can ask questions to encourage you to develop more multidisciplinary projects. Let me go through a few of these, a few of the highlights we have here. We have a large core instruments run by Sergei Nikonov. He runs patch clamp systems, micro electrode array, and confocal two-photon systems here. These are some images of IPS-derived retinal ganglion cells transfected with different markers and stains, so you can see the level, the quality of the images available with the confocal system. In this image, you can see they are EGFP expressing ganglion cells that have been derived from the IPS cells. And on the left, you can see the fluorescent image. In the middle panel, you can see the bright field image of the patch clamp electrode going on to those neurons. And in the right panel, you can see traces from the current clamp mode of patch clamping. You can see on the top, there are spontaneous firings, and on the bottom, the spikes in response to depolarizing current. And so this combination of the microscopic imaging and the patch clamp is really important when you want to ask questions about function. In this question, are these cells responding like ganglion cells? Are they able to initiate an action potential? And again, these are from the Jawali lab. One of the other aspects of Sergei's work is to help with the microelectrode array, and you can see an image on the right of a piece of retina in a microelectrode array, and this again combines electrical recordings with light stimulation. You can see in the panels that we have light being produced with these little bars, and when light goes on, we can see action potentials coming out from retina through the field. And this is analyzed, so we can see that there's a big increase in spikes at the beginning of light onset, a slightly smaller increase in firings when light is turned off, and there are some neurons that fire throughout. And so the MEA really is a very powerful technique if we want to be able to look at how the output of the retina, the ganglion cell spiking, can respond to light. And as we develop new techniques and ways to test this, this can be useful, and we've been useful seeing it applied to some more clinically relevant projects recently. And I just wanted to point out that the visual system isn't just about the eye, and so I thought it would be important to just briefly show an image of Sergei's recording from the brain, because I think we need to remember the brain is very much a part of the visual system. And so this combination of electrophysiological recordings, stimulation becomes useful when we're looking at other aspects of the visual system. Now, we recently had Brent Bell join our module. He an expert in looking at small animal imaging resources and capabilities. And so he uses techniques that are available in the clinic, OCTs and confocal SLOs, to image in live animals. He has prepared an excellent PowerPoint presentation that goes into the details and also teaches people. So I'd encourage anybody interested in this to reach out to Brent to receive this and talk to him. I've included a couple of images here just to highlight this. For example, these are images from his presentation from the Deneyev lab, showing how he can use the OCT to be able to visualize changes in the thickness of the retina. And you can see on the left, there is thinning of the retina in response to challenges. And to be able to measure these changes in a live animal is enormously important, particularly as you want to look at time courses in response to the damage. We also have the confocal SLO to be able to image the machine. You can see this on the top right, the image of the system, again, that was recently awarded as part of a shared instrument grant. Showing below that just images, I think again from Josh's lab, showing the different wavelengths of reflectance. That blue light is reflected at the layer of the RPE on the top, and we can see through the choroid using a higher wavelength light. And so this allows us to be able to image different modalities. I've included on the right images that Brent just gave me the other day, in which we're looking at the retina of cells which are fluorescent, the CX3CR1 GFP cells, in which the microglial cells are fluorescent. And the top is a control, the bottom is showing damage that happens in response to our chloroquine retinopathy model. And one of the reasons why I wanted to show this is that, Brent is very responsive to the fact that we have a grant due. And I think, we all like to use these facilities to help us generate data for our grant applications. And it's great that everybody involved tends to be responsive and aware when you have deadlines. And so I thought, it's important to stress that. We also have a Nikon A1R confocal system at the dental school with Kathy Battaglia and Anu Dinga. And just some images here, beautiful images of the RPE, and you can see the orthogonal structures here. On the right is an image made in conjunction with collaborations at the vet school, showing incredible imaging of some of the issues we see with best stroph and the best disease there. So the quality of these images becomes, very important. And again, generating images for grant submission or publication. We also have been able to interact and support the Vet Imaging Core. And these images you can see of the HEK 293 cells transfected with a GFP tagged VP40 protein. So again, we offer a discounted usage at the vet school and the dental school for the acquisition of these high quality images. So I hope this has given you some idea of the options that you have. We have an ability to really transcend the quality of the imaging that you're able to do, but also combine this with electrophysiological measurements that are critical if you want to show changes in function as part of either a normal visual process or pathological extensions. And now with Brent being able to extend this to the live animal, small animal imaging, really gives us a lot of tools to help with your research. So I'd encourage all of you to just think about the questions that these tools allow you to ask and talk to these experts. And I'm sure, they're very accommodating and can tell you what will work, what won't work, but use your imagination and take advantage of these incredible resources we have here.
Great, thanks, Claire. I think next up is William Beltran, who will tell us about our instrumentation module and its components.
Yes, thank you, David. So the core instrumentation module is really here to I would say, to make your wildest instrumentation dreams become reality. And this is done by basically giving you access to top-notch machinists, top-notch electronic engineers who are not only able to produce, in fact, prototypes of the instruments or machines that you need, are also able to reverse engineer any machines that may be commercially available that doesn't fully suit your needs and that you need to adapt to the specific experiment that you have, and also to repair any machine in the lab that you may have and that is non-functioning. So what I thought I would do today, and there's no better time than when we're confined to our respective homes, is to take you through a stroll through campus, meet Freddie Letterio, meet Miguel Hernandez, and they're going to take us to, in fact, their shops, the machine shop and the electronic shop. So just bear with me, and I will start this short video.
I'm Fred Letterio. I'm the manager of the medical school section and our machine shop at David Rittenhouse Labs. I have Ben Franklin behind me and College Hall in the distance behind. And so I'd like to show you the best way to get to our shop from this point. So we want to go east on Locust Walk, and we'll take a little tour of that right now. And I thank you so much. Bye. All right, so we're on Locust Walk here. Okay, we're on Locust Walk and 33rd Street, heading north to our building. This is the David Rittenhouse Labs, and I will be meeting you at these entrance doors right here in the front of the building. This is Fred Letterio. Once you reach the front doors, come right on in, and I want you to call my cell phone number, 215-356-3705. Now we're going to go up through the lobby and down the hallway to our shop. Okay, so actually, here we are at the shop, and I'll show you the office, my office here, and short little introduction to our guys. Mr. Mike is Mike Suplick, right-hand man, fantastic machinist. Here is Jeff Hancock. He's the genius behind all these computerized machines. We'll come over and see if we can find Buddy. Hey, Buddy.
Hello, Mr. Fred. How are you?
Expert machinist and welder. Hi again, Freddie here. I'm going to show you some of the jobs we've worked on, sketches and photos, for the Mahoney Institute of Neurosciences. This is a sketch somebody brought in and said, make me these. Take sizes, make a sketch, and put the sizes on it, and then you can actually build that. This is another sketch for the same doctor, Dr. Dave Brainerd, high-resolution imaging display system to improve the accuracy in size and distance, perception, and stereoscopic displays. Okay, so that is that. We'll go over to another. This is a TMI assembly, traumatic brain imaging. And this device holds the TMI on the other end, goes around the skull of the patient. We made 3 of these. I made 2. Michael did a beautiful job on the 3rd one. That knob, the red shaft, rotates. The TMI device rotates around the skull. They can take readings from that. Over here, we have a dog wine maze. And it's a little write-up on it says protocols for visually guided navigation assessment of efficacy of retina-directed cell or gene therapy in canines. Again, this was for Dave Brainerd. This was also William Beltran. This was Gus Aguirre, Jeff Aguirre, and a host of other vision doctors in our group. Yes, I'd like to give you my phone number again. It's my cell phone, 215-356-3705. And my email is Letterio, Letterio, at pennmedicine.upenn.edu. And thank you so much for your time.
Good afternoon. My name is Miguel Hernandez, and I'm going to show you Penn EDS, the Penn Electronic Design Shop. We are here at the Locust Walk next to Ben Franklin, and we're going to take you to our lab. So Penn EDS, the Penn Electronic Design Shop, is on room A15. This is where you can find us. This is in Solomon Labs, and I'll show you inside now. So first we have the main office, I guess, where we meet our customers and we decide what are the needs of the researcher. We are basically focused on value. We provide value to our customers on their research needs. We are in the job of solving their problems, of coming up with solutions in the electronic design area so that they can do their research. So we're partners in their endeavor, to find new discoveries. Go and see what we have, the equipment that we have. We have everything to build your prototypes. This is where Alex works to own your schematics and your PCBs. This is the area where we work with the prototypes, where we come up with components in order to make and build the solution for your research. We have cables, we have capacitors. We provide solutions to our customers by using all this equipment. We now go to A16 where we have a small machine shop. We have some tools here so we can make those boxes so we can enclose the electronics inside. We have an oven, a wave oven, where we can cook the PCBs. After you put the components, the surface mount components. There's a 3D printer here, the saw, the drill, some basic tools to make the necessary changes to aluminum boxes so we can enclose your electrical project.
Hi everyone, my name is Alexander Santos. I am an electrical engineer here at Penn EDS. I am glad to show you some projects here at the Electronic Design Shop that we are working right now. For example, this is a box for Duran Lab. We are going to have 6 boxes in here and panels, LED panels in each wall. It's going to be light everywhere, and we are going to make a controller here similar to this one. And this is a controller for heat induction. And with this controller, you can control the time of operation of the induction heater, and also monitor the voltage and take action independent of voltage and the time. And with this project, we are going to have something very similar. Instead of having a safety feature with voltage, It's going to be the same but with temperature. We are going to have a time controller to control the time of the LEDs and the intensity of the LEDs.
OsiFlex is the first medical device designed and produced here at Penn EDS. OsiFlex is a startup. This is how we implemented and tested, validated the final prototype after years of development. You see the leg. The prototype started like this. This is the very first design of such prototype. You see the protoboard there in the pneumatic system. So from that idea, we went ahead and developed a PCB.
After that PCB was designed, we integrate the pneumatics and the PCB in Fusion 360 to come up with a design. After that design, this is how it looks like the internal of that design. And this is the best setup for data collection and characterization of the OsiFlex system to make improvement for the next iteration.
The next jewel is a VRC project funded for Dr. Jeff Aguirre and Dr. Eric Kaiser. This is for stimulating mice at different wavelengths and different flickering times. You see a MATLAB program controlling 4 boxes with 3 channels on each box with UV light, red light, and blue light. And we have a main box that is controlling all those channels.
So I thought that this gave you kind of a snapshot, an idea of the type of expertise that you have truly on site. And I would just want to emphasize that the individuals you saw and their teams are really people who are invested in understanding, first of all, what is your science, what's the question that you're asking, and I think that although it may seem as a black box, some people don't realize that you really have to walk in, have a chat with these fellows. You need an idea, you need a very, very simple graphic, and they'll take it from there. So that's wanted to thank, in fact, Freddie and Miguel again for having prepared these videos, and hope you get in contact with them. Thank you.
Great. Well, thanks both William, and then for Freddie and Miguel and Alex for the multimedia extravaganza. That was great. I will mention also that the shop's instrumentation module does charge hourly for its services, but the VRC subsidizes the labor to quite a large extent, so that cost to you and your lab is well below market price for vision research projects. Our final module will be presented by, which is biostatistics, will be presented by the module director, Guishuang Ying.
Okay, so everyone can see my slide. Let me show the full screen. Sorry. So I want to talk about the biostatistical core module, and I will cover who we are and what we can do, and how you can proceed if you need help from us, and where you can find us. So we are a small group, and I kind of the leader for the biostatistical module, and I have worked in this module for over 20 years. So I have some experience in biostatistical consultation for the ophthalmology and the vision research, and analyze the data from ophthalmology and the vision research. And I also have a few master level biostatisticians support me to do the actual work. Sorry. And we have Yinxi and Brandon and Peiying, and they are master level biostatistician, and we also have a biostatistician left us last month, and we are hiring 2 replacement. So we have 4 master level biostatisticians work for the fishing call grant. They are not working all the time in this project because they have a lot of other projects which is outside of fishing call grant and keep them busy. But everyone get involved some of the project for the fishing call grant, so, because this project is good for them to have some exposure on the ophthalmology and the fishing research. And what we can do, so I think we can do in many things, and even start with the study design, either in the laboratory research or clinical study. Certainly we can help with the power sample size calculation. Probably everyone know we can do that. And sometimes we can also help to select the study outcome, primary outcome or secondary outcome. And we can also help you kind of design a data collection form and also design a database. And certainly we can help you with the data management and the statistical analysis and how to interpret the analyzed results. And when you have a result, we can help you to write the statistical analysis session for the paper. And finally, we can collaborate with you for writing a grant application for either the R1 grant or the other type of grants. And for the statistical analysis side, we can do a lot of different things. Certainly, we can do some basic statistical analysis. Probably most of you know, we can do a t-test, ANOVA, linear regression model, chi-square test, logistic regression, survival model. And more importantly, we can do a more complicated data analysis, particularly in the ophthalmology and the vision research. You know, every subject, most of the time, they contribute data from 2i, and we have to somehow account for the entire correlation. I will go a little deeper in the next slide. And we can also help for analyze very large data and just like the medical claim database and electronic health record data. And this kind of data is more complicated because they always have a lot of confounding factor you need a value for, and they have some specific statistical method you can use. And we can also help you to evaluate a kind of diagnosis test, see what's the sensitivity, specificity, and the area under the curve analyze. And finally, my personally, I have a lot of experience in a kind of development for a prediction model for the eye disease and how to validate the prediction model. And in terms of the statistical package, we commonly use SAS, and we also can help with use the other software like R and the Stata. The one thing I want to kind of bring to the attention is that we are trained as a biostatistician, not a bioinformatics. So we don't have a lot of experience in the surface for bioinformatics because the data is much more complicated. It's very computation intensive. But if you want to kind of ask help, and you can reach us, and if we see we are not comfortable to do the help, and we can point to you where you can find the help. And I already pointed out in the eye research, we always deal with correlated eye data, you know, because most of the time we easily can take the measurement from both eye, either in the human or in the animal. And the data from two eye of the same subject, they are often positively correlated, and the statistical modeling, we need to account for the entire correlation. If you don't account for the entire correlation, you may lead to the invalid conclusion. And I review a lot of paper, and this is a very common mistake people often make in ignore the entire correlation. So if you have a kind of correlated eye data, you are not sure how to deal with it, I always encourage you to come to us, and we can provide you the advice. Either we can help you do the statistical analysis, or we can point to you or how to do exactly by yourself. And in the past few years, we know the correlated eye data is complicated, and a lot of people, they are not aware of how to analyze the correlated eye data. So we develop the time to develop a few tutorial paper I listed here, and the paper cover a lot of different topics on different type of data. The first paper, for example, is a correlated eye data for the continuous outcome measure, like the visual acuity, intraocular pressure, which is a continuous measurement. And the next paper is correlated eye data for the binary outcome, just like a disease, yes, no. And we also have a tutorial paper on how to analyze the diagnosis test using the sensitivity, specificity, predicted value. And we also cover the correlated longitudinal data, eye data. Basically, you not just measure the data at one single time point, you measure the data at multiple time point data, which is another layer of correlation. So it's much, much more complicated. And finally, we have a tutorial paper how to do a kind of RC curve analysis for the correct eye data. So if you have a project, you need us to help and how you proceed. I think the best way you start, just send me an email, I give me a phone call, and say, then I will reach you back, and we may want to schedule a time to have a meeting, either by virtual. Now we only can do virtual, but I personally prefer the face-to-face, so that you can tell us more about the project. What's the study question, how you collect the data, what's the data look like, and I can provide the best advice how to proceed. And a lot of time you may want us to help the statistical analysis, and you give us the data, we can do the actual statistical analysis. Then after we have the statistical analysis results, I will schedule another meeting and go through the results with you to help you understand the statistical analysis results. And you may have some additional comments, feedback, and we may need to modify the statistical analysis. So we can back and forth a few more times, and finally we get the final analysis results, and you write a paper, and we can help you write the statistical analysis session, and we review the paper. We think it's good enough, we can ship out to the journal. So this is the statistical analysis part. And you may wondering, do you need to pay us? The fishing call grant cover our salary, my staff salary. So I should say no charge unless the project is really huge. Project need us a lot of time, and we can talk if it's the case. And how you acknowledge our contribution. Certainly, if you think we make a large contribution, it's worth authorship. You can be put us into the co-author. If you think our contribution is minimal, you can just acknowledge us in the paper. And for the future grant application, if you think we need provide you a lot of support, you can consider put us some percentage for the salary support for the new grant application, like our one grant. And I just, from my experience, I just put a few notes so that you can be aware of. I think currently, because we are busy with a lot of projects, so the process I traditional use is really first come, first served basis. But certainly, if you have a grant application, have a specific deadline, we certainly can put this a high priority. And I also encourage you to approach us earlier, even at the study design stage. Sometimes we can provide you some good advice and try to avoid some mistake, And you may wondering how long we need to spend the time to the work. I usually encourage you, if you have deadline, you should approach us about 3 to 6 weeks before the deadline, so that we have plenty of time to do the work. And I encourage you not to advise you not to do the last minute request, particularly before our abstract deadline. We have a lot of projects working on, and sometimes we really cannot deal with the last minute request. And for the study, we are not responsible for the IRB approval, and you should include us for IRB if you want us to do the statistical analysis. And when you have a project, we provide a service. You should keep us to updated on the abstract for the conference, publish the paper, or funded grant, so that we can keep track of our contribution, and this will be useful for us for the grant renewal for the fishing call grant. And certainly, you should acknowledge the fishing call grant funding P30 for the paper or for the future the meeting, presentation, this kind of stuff. So how you can find us? We are currently we work from home remotely, but our address is 3711 Market Street, Suite 801. And here is my phone number and email address. That's it.
Great. Thank you very much for that overview. Let's see. So I think if now is certainly some time and we can hang out a little past the hour if there are any questions from you guys out there, either about the modules or the VRC, or we're happy to take them. You can unmute and ask or type them into the chat. While people are thinking, I'll mention that Jeff Aguirre had to run, but he says he's happy to talk to anybody about how to get your data onto Flywheel or the Flywheel system more generally. So just reach out to him in that domain.
David, I just want to thank you for your service and all the module directors for these really valuable resources.
Thanks, Josh, and thanks, Paul, for the nice comment in the chat. We don't have a question. Do you have an illustration core? We don't have an illustration core anymore. Mary Leonard actually has left Penn, but she is working freelance. And if you have specific work you'd like done with her, check with me. We've been continuing to sort of do our previous 50% subsidy of her hourly rate within reasonable bounds of the scale of the project. I believe she's still available in the Philly area for that. And I guess even if she's not in the Philly area these days, we've all gotten good at remote. So that's still available. Well, okay, I'm not seeing any other questions, but you have in the chat the handout sheet and the web URL for the VRC where the same information is available. And as I said, what we really want to encourage everybody to do is be aware of and to use the core modules. Our product, as it will, or our specific aims are to deliver services to the Penn Vision community. And we are really eager to do so and to help all of your great research proceed. So with that, we'll close today's session. Perhaps see you all at the next substantive vision seminar, and have a great week.
All right, thanks everyone.